All right, here we are again. Let's see how the videos are going. That looks good. And this is going. So what you're watching is the Brachypodium diversity set. This is four different chambers. Well, four cameras in two chambers. The ones on the bottom are simulating a warmer climate than the ones on the top, and we're watching the grass grow. So that was a season in the bottom, and the top you can see the lights. This is going through one diurnal shift. So we're investigating the green pixels across time as a phenotype, as a quantitative phenotype. It's a bit messy at the end, of course, but different SNPs play a different role in controlling growth. And so this video is looking at different days, the number of leaves on different days, and across the x-axis at the bottom is 5 different chromosomes, and those are all the million variants that are segregating across the 5 chromosomes and the 100 different Brachypodium Distachian diverse successions. And then each point is on the y-axis uh, the test statistic between a SNP and the growth pattern that particular day. So essentially you can watch the gene action come on and off through development and across environments. There's a big gene in the middle of chromosome 2 that's important early on, and then genes on chromosome 3 come up and down later. And so this is one of the first dissections of the complex trait of growth across environments and across development. And you can track it down to essentially a handful of major genes that have different roles and different conditions at different stages. So when you reverse these models, we can then make predictions of growth and development. We wanted to do this in field conditions like Wagga Wagga, New South Wales, where it's a major wheat growing region. We have thermal conditions, and instead of counting development in the number of leaves or in days, we're also thinking about degree days. And those of us who have been outside realizing that spring is rolling in a major way, you get so many more hours of good growing conditions in spring and summer than you do in the winter. So treating every day equally is not the best way. This thermal time is a cumulative degree days over the season, and that's how you can see the warmer future climates that we're simulating for this 2050 here are really the cumulative conditions come in much quicker than be expected. So we've got other tools for phenotyping. This is a whole omic science in and of itself called phenomics, which is just trying to get measures of traits automated and through time and across environments, standardized. Chlorophyll fluorescence and thermal respiration. There's different, um, the way leaves pass water and transpire, that cools them off, and you can look at that in the infrared image. Greenness is related to fluorescence, and we can look at chlorophyll fluorescence using a pulse modulated camera. And so these are all different traits and measures. And if you have a quantitative number on every one of your plants and you have genetic variation, then you can test whether this phenotype is actually heritable and is it due to a certain number of genes. So essentially, we throw all the tools in your lab at the phenotyping once you've got a core set of lines, and then look for the genes to come out. So seedling height and leaf 3, those phenotypes are very similar in the future conditions versus the present conditions. And many genes control those traits. Now, there's a few data points in this slide. Some of them are talking about like respiration per leaf area. So if you clip off a leaf and put it in and look at how much it's breathing, how much oxygen is consumed by that leaf, then there may be a few suggestive genes in here, but it's largely below the threshold. And this threshold is assuming that every test is independent, which is really a conservative threshold. And the y-axis is different on all these plots as well. Seedling height is more of a developmental trait, and it seems to have a few more genes in here. And then the relationship between growth rate and input, photosynthetic efficiency, and output, respiration, and should result in growth. And so we call this energy use efficiency. This is just an example of how you can play with a lot of different traits through time and across environments to look at which ones have the highest genetic signal. Start breeding for those new traits. And then you can go down to the genes and say, well, what is this? And there could be a transcription factor, some DNA binding protein, hormone response, nutrition, phosphate related responses, and try to get at the mechanisms underlying this. So this is the Whirlwind Tour. So you might have heard of genomics certainly before, phenomics now, and vironomics is even something that being launched in a big way over at CSIRO. And this is a bioinformatics class, so the tools are about integration. And this is called systems biology or even systems genetics, to try to look at development in a dynamic environment. You have these different layers from the genome to epigenome, transcriptome, metabolome we haven't talked about, but these are destructive approaches. Or spectrome there's spectranomics. It's another omic where you look at the multicolor spectral images and try to predict the leaf chemistry or the physiological state from that. And then those predictions can themselves be phenotypes that can be related to genotypes. So you need to be a data analyst. You need bioinformatics across this whole sphere and be able to track your data points, have good experimental design, and process big data. So the tools are often quite simple. The statistical tools, just a mixed effect model is the only thing we're doing here that accounts for both populations and SNPs and multi-trait approaches. I think the challenge is to have good experimental design and get all the tools working, and then make predictions from that. And that's the state of the art at the moment. It's exciting times for biology. We're seeing engineering and computer science merging into biology. Biology, we have some of the biggest problems of food and environmental security, and we need all the solutions that we have. So this genomic approach to linking phenotype and environment has a really big applications in the world. So it's a good team, and I'm happy to be able to present this to you guys today. Thanks a lot.